Hello, I'm Chang Lin, the co-founder and CTO of EEST. Thank you all for attending today's webinar. As we previewed in our previous webinar, today's topic is drug development and AI, specifically focusing on the possibilities of foundation model. So today we will cover the following topics. The first part is what are foundation models? Then we will look at 5 reasons why foundation models are necessary for drug development. Then we will examine how these foundation models and discuss the present and future of foundation models in sequence. After these main topics, I will have a Q&A session via chat. For our first topic, what are foundation models? Among those attending online, some may have heard of foundation models, while others may not. So for those who haven't heard about them, let's examine what foundation models are step by step. So when we talk about AI, we typically have a problem like collecting relevant data and training on it and make predictions in these stages. Among various types of AI models, a foundation model specifically refers to an AI model that can comprehensively learn and understand large-scale data from various domains rather than specific to a particular problem. So while typically AI models, as I mentioned, are trained to solve specific problems, foundation models don't focus on specific problems but process multiple types of data simultaneously. For example, if there's an AI that can classify or summarize documents, it collects trained data related to that task and learns to predict based on that data. And if you want to create an AI that generates music, you gather music-related data and train it to predict. This is how conventional machine learning or AI works. In contrast, foundation models take all the data using individual tasks and learn from them collectively. So naturally, the amount of data increases and thus domains become more diverse. The biggest advantage of creating AI in this way is that it shows high generalization performance across various domains. Of course, using domain-specific data might solve the particular problem better, but for example, if an AI model is trained to summarize documents, that model cannot be used for music generation. So foundation models are good because they can be used across various domains. And based on these trained foundation models, true to their name as foundations, if you want to apply them to a new domain that the model hasn't been learned yet, you can collect a small amount of domain-specific data and achieve high performance through so-called fine-tuning. This gives us the advantage of being able to apply the model to domains you haven't trained on. So one of the most remarkable aspects of foundation model is their emergent intelligence. Large language models like ChatGPT, Gemini, or Llama, which are representative foundation models, have been trained on enormous amounts of data from various domains. As a result, they show unexpected capabilities. When we say unexpected, it means that the model shows behavior that weren't explicitly specified or programmed by the people who created it. For example, here's one case: asking for jokes. When asked I want to hear a joke about Madonna, GPT 3.5 responds with something like, "Why did Madonna go to the bank to get a material loan?" It's not very funny, and it's more like a fact. But however, GPT-4, trained on much more data compared to a previous version, says, "Why does Madonna study geometry? To learn how to strike a pose from every angle." And it's become better. So it creates questions and answers by itself to make jokes. Recently, it's been noted that these LLMs can also reasoning. For example, when asked which is better for passing through an infinitely wide entrance, tank or car, Previous GPT version answers both could pass through, but consideration of entrance height and depth is needed. Meanwhile, GPT-4 responses, since it's infinitely wide, meaning there's no width restriction, both can pass through. Up to here, this might seem reasonable. But then I capture this image, and this is an image, and fed them directly to GPT and asked it to explain the image. And here's what GPT is explaining. This image shows example comparing the performance of GPT 3.5 and 4. It's divided into two cases, a joke case and a reasoning case. It then repeats the content and provides an evaluation. 3.5 just presents a joke, 4 presents a more creative joke. This is GPT-4 providing commentary. It can do things like this. Then regarding the reasoning case, it notes that while 3.5 provides conditional answers, version 4 provides simpler and clear logical answers. This isn't my writing, this is GPT-4 providing this evaluation. It shows much better reasoning. Adding to this, it provides this conclusion. As can be seen in this image, GPT-4 provides more creative and logical answers, showing improved responses compared to previous GPT version. These capabilities weren't explicitly programmed into GPT-4. Moreover, when given the text contained in the image, it was asked to emulate this situation. It accurately understands the content from the image and provides appropriate commentary. This is an example of emergent intelligence. These are the good points of foundation models. They generalize across various fields and show emergent intelligence. They can perform new roles that weren't expected. Students in our lab also make extensive use of emergent intelligence of these large language models. For example, when they read papers, if they don't understand some mathematics, they can capture that slide and say, please explain this formula, and AI explains it. Going even further, if they say, I want to go from here to here, but I don't understand, then the derivation process completely. Can you explain it for me? Then they explain the equation. It's developed tremendously. And if they say, I want to make this equation into code, could you write the code? And the GPT writes code too. It can perform various functions like this. So based on this, let's look at why foundation models are necessary for drug development. Into 4 or 5 reasons I've identified. I can summarize into these 5 points. First, data integration, management, and analysis becomes possible. Second, we can overcome the limitations of insufficient individual biodata. Third, precision medicine becomes feasible. Fourth, we can create explainable and interactive AI. And finally, we can implement emergent functions that we hadn't expected. And these are the 5 reasons why we should introduce or apply foundation models to drug development or bio research. Looking more closely at the first point about data integration and analysis, as we discussed in our previous webinar, biodata itself is very heterogeneous, and even within a single cell from the molecular level, these molecules including DNA, RNA, protein, and etc. They are extremely diverse protein molecules, and their quantities are heterogeneous and hierarchical. These interact with each other to show highly nonlinear reactions, and consistent and orderly patterns exist within them. These are what we need to deal with in bio. So individual data, for example, does understanding DNA alone as understand cells. So we need collect all the data into and try to understand really comprehensively. So it's impossible to understand the whole phenomenon by relying on individual data alone. These are characteristics of biodata. To solve this well, we need an integrated approach through foundation models that can utilize various domain data simultaneously. Looking at this from a different philosophical perspective, so-called reductionist approach, which I talked about in the last webinar, we tried to understand bio from a reductionist perspective. The same goes for AI models. From an AI model perspective, these are called model-centric AI, which means there will be various forms of data that can come from bio and there will be various problems. So we've been collecting large amounts of data for specific problems and training them to solve the problems we want. Like the task-specific traditional ML I mentioned earlier, that's how we have approached bio problems. But as I mentioned on the previous page, just because we can predict individual element structures well, predict gene expression data well, and predict drug response well, it's not enough. It doesn't mean we understand the whole well. However, to develop drugs well, we need to understand the entire cell or entire tissue or entire organ to get good results. The reductionist approach is problematic from the start for approaching in this way. For example, even though AlphaFold can predict the protein structure, and they predict it and make a database for the all existing proteins, nobody says we understand life phenomenon because of that. Even understanding just individual cell unit activities isn't really solved by knowing this. It just ends at knowing the structure. In some ways, this might be an example of proposition that the whole is greater than the sum of its parts. In other words, it seems there are clear limitations to predict this emergent phenomena, this unexpected new phenomena that arise from the combination of each part using a reductionist approach. Put differently, rather than reductionism, perhaps we should approach bio with a holistic method, or what we call holism. That should be how we approach it in the future. And from an AI perspective, this is called data-centric AI. So we gather all these individual data types, structure, gene expression, protein, drug response, and I learn them all together comprehensively. The learning method isn't about solving specific problems, but developing AI models through unsupervised pre-training. And going further to understand the interactions between these data from different layers, we need to understand their correlations more comprehensively. Then for the individual tasks we want to solve, match structures, match gene expression levels, and this individual task can be better matched using small amounts of data through zero-shot or fusion learning methods. This is the first reason why foundation models are necessary in the bio field and drug development. And the next reason is overcoming the limitations of insufficient individual bio data. This graph, which I showed in the last webinar, show how rapidly single-cell sequencing data increases over time. Looking at this graph, it's on the y-axis is on low scale, and after 2090, it increases, roughly doubling every 6 months. The data is accumulating at an incredibly rapid pace. The data being collected is already about 14 petabytes. However, the researchers say that this isn't enough to simulate a live cell. So no matter how much individual data we collect, it's not easy to explain a living cell. And in many cases, we can't even collect this amount of data. So we need foundation models to overcome this. As I mentioned, individual AI models For example, if you want to make a model to predict potency, you collect activity data for specific proteins and choose a model like a graph neural network to train and make predictions. While we have done it in this way, if we apply foundation models, it's not just about activity data, but we can utilize all kinds of basic data that can serve as background, general biological data, chemical data, mathematical data, physics, pharmacy, medical data, and etc. We train using all these data, and after training, we fine-tune the model based on this trained foundation model for the specific protein activity data we want to solve. Then this model can more clearly define what activity data means and what target proteins mean, and with the background knowledge it has, it can perform individual tasks like potency prediction better with just a small amount of data. So this is what we call data-centric AI, and with such good foundation models, so-called zero-shot learning or fusion learning becomes possible. Zero-shot here means predicting without using any data for a specific problem, but just using a pre-trained information only. This becomes possible with foundation model. I will show you an example of this later. Fusion learning means high performance prediction is possible with just a small amount of data for a specific problem. Literally few shot, meaning with just 3 or 4 examples. So in fields like drug development or bio that are so-called data hungry, where collecting data is really expensive, these zero shot or few shot techniques will be very critical. Next, I will show you one example. This is an example of drug repositioning. This is a very recent paper in Nature Medicine. So it doesn't have any volume number yet. What this paper tries to do is drug repositioning or drug repurposing. According to this paper, roughly 17,000 diseases they studied, and 92% of them have no indication. Especially for rare diseases, 95% have no FDA-approved drugs, and 85% don't even have drugs in development. So in this situation, we can do repositioning or repurposing. That's what this paper discusses. In this paper, they integrated biological knowledge represented in graph form for pre-training and created what they named as type of foundation model. They named it TextGNN. And after creating this foundation model, they show that repositioning is possible even for conditions with no indications and no previously developed drugs. This is the model's form, and they represent each piece of data as a point and manually indicate the relationships between these data and other data. They call this the graph or knowledge graph, and they created this knowledge graph and mapped countless data points to it and pre-trained the model with the graph. Then based on this, they do later is input the disease and drug you want to match and find out if these two are related or not related. So these people used hundreds of millions of biomedical data points for pre-training and obtained all the embedding nodes that were passed as points. And we run the relationships between them and finally created a foundation model. Here, their foundation model, and the others are models trained on individual tasks. Previously, people made deep learning models for drug repositioning, and they compared the performance for two cases. The first case is when the test disease have the treatment in the training set, and the second case is when the test disease don't have the treatments in the training set. For the first case, some models give the quite high values compared to the model in the model of the paper, and they are close to 1 on the AUPRC metric. Similarly, foundation models also gives high performance. Similarly, it gives so we can conclude that both models give similar or better performance. But for the second case, the below result is different. There are no drugs for this disease and the trained data. When asked if this can be predicted while existing models cannot predict it well, the foundation model still shows high performance. Since there's no data, this becomes zero-shot learning and it can predict even without any drug data for this specific disease. This is one of the good performance of foundation models. And extending this further to cases with no indication, then completely new disease, meaning no data at all. And the problems are there is a disease, but the related biological data is very sparse. When they tested on various indications related to autoimmune disease, those trained on individual tests naturally show low performance because they don't have the data to measure similarities. However, the foundation model still shows high performance. It means that the foundation model shows high performance in the fields with a very little data. Foundation models can be a very good solution in such data-hungry fields. Third, we could realize precision medicine. Precision medicine is ultimately about creating some form of personalized medicine because individual people have different genes and their habits based on their genetic type. We need to make personalized prescriptions accordingly. The problem is that the data we can get from a specific individual is too limited. It's impossible to solve this problem in a data-intensive way. But if we can utilize zero-shot or fusion learning techniques, as I mentioned earlier, a naturally precision medicine becomes possible. This will be the third reason why I need foundation models. Taking this further using artificial intelligence to create virtual cell models. I introduced this in more detail during our last webinar. If we have just virtual cell models that learn by integrating diverse data from molecular level to cellular level, we can select an individual's genotype to create personalized cells and use them for precision medicine predictions. All these things should be possible through foundation models. And the fourth is that we can create explainable and interactive AI. As I showed in an example from the Nature Medicine paper introduced about repositioning, they show that instead of just making prediction like this, you can communicate with artificial intelligence like this. If you ask this model to suggest potentially repositionable drugs for disease A, it can suggest drug D might be good. And when asked why this drug is more appropriate for disease A, it provides an explanation. So there's a separate AI attached that provides explanation, and although it's hard to see here because it's small, it shows the mechanistic pathway in the knowledge graph for why drug B connects with disease A. So people can look at this pathway and evaluate whether it's good or not. It's possible to have AI that can explain and interact with human researchers. And for example, when I asked to reposition for a specific syndrome, for example, in this paper's examine, Dorpidem came out. When asked why Dorpidem fits well here, it gives specific answers, and when people look at these answers, they find them reasonable so they can evaluate them experimentally. In this way, explainable and interactive AI implementation can be achieved through foundation models. Finally, emergent intelligence can be implemented. This is literally unpredictable, but it's talking about intelligence that might emerge even though we didn't intend it. For example, I'm giving this as an example since we haven't seen anything like this yet. In existing drug development, you find targets, find cases, and go through hit to lead and lead optimization process before going preclinical and clinical trials. It goes step by step, and each step requires appropriate data or test. For example, if biologists do target identification and chemists do the subsequent process, they make molecules. Then people with expertise in prediction do those parts, and finally doctors do clinical trials. So while everything is divided by stages, if we have a foundation model that has learned all this collective data, we could do drug development that considers everything from target identification to clinical trials simultaneously. So no human knows all these fields simultaneously, but if we can digitize all this and AI can connect to all this data, we could go to this end-to-end stage where even when finding hits, we are already considering clinical trials while finding hits. This might be what people want most in terms of emergent intelligence. So to summarize what I have talked, LLMs are representative foundation models. They've learned an enormous amount of data in various languages, even learning ultra large scale data beyond what humans can digest. As a result, they can do amazing things, breaking down language barriers, solving mathematics, as I mentioned earlier, understanding situations, and these days, LLMs are even said to be used in operating humanoid robots. They can perform such diverse tasks. In bio, the data itself is unbalanced, unstructured, and hierarchical. They transmit information between themselves, like the central dogma information transfers that cascade from DNA onwards. In some ways, this is like natural language, but it's much more complex than natural language. However, in terms of information translation, is very similar. From this perspective, just as LLMs showed great success in natural language, if we can create foundation models using these various forms of data and information flows, just like we achieved great success in LLMs, we might be able to utilize artificial intelligence well in fields like precision medicine, rare disease, and drug development. Next, moving on to our third topic, let's look conceptually at why foundation models work so well and how this is possible in a short time. First, there are three elements that make foundation models work well. The first element is having lots of data. Big data is available because it's not for individual tasks. We collect all the data we can collect. Because we are not just solving specific problems, but gathering all available data regardless of what problems we are solving, the data size is naturally very large. We are dealing with hundreds of billions or trillions of data points. For example, for LLM cases, we collect all data that exists in language. Whether it is in Korean or English, we collect it all data. So how do we process this data? A key way of data processing is self-supervised learning, what we call self-guided learning in Korean, which utilizes unlabeled data. For example, in natural language, let's say we have this sentence, I really like hyperlap made by him. And we do random masking into the sentence. For example, if we mask it to I really like blank made by him, this AI knows what the answer is, tries to predict what goes in the blank. And since we can do this randomly, we might mask I really like hyperlap made by blank. We can predict both parts. Since we can do this randomly, we can produce enormous amounts of data from even with only one sentence. Because it enables self-labels and self-learns, we call this self-supervised learning. For images, as shown in the present slide, we do masking like this. We mask specific parts of image and fill in the blank spaces. We want to learn protein sequences, we can mask sequences, and the model is trained to predict these empty spaces. In this way, we can create countless data points even from one protein sequence. This is the really important aspect of making a large foundation model. And the second important aspect is ultra large scale models. Naturally, as data increases, the complexity must increase to understand the complex relationships between these large data. We usually talk about this in terms of parameters number, and this graph shows how the number of parameters has increased over time. This is on log scale, and while in 2017, once the model size, the model is on the billion scale, it's now grown to tens of hundreds of trillions of parameters. The model's complexity has become incredibly complex. So artificial intelligence models have been created that can learn enormous amounts of data and learn the relationships between them. And another important axis is the computing power provided to train these massive models with incredibly large amounts of data. And this is the biggest reason why NVIDIA makes so much money. The graph on the right shows how gradually GPU bandwidth has increased on a low scale. Both are increasing on a low scale. That's how foundation models can be created. So let's see what effect does the self-supervised learning give. Through this, we can identify patterns, but for example, in supervised learning, you are given specific problems, but here problems are not given. We create problems by ourselves. By doing masking like this, rather than answering predetermined questions, it's like studying independently without going to an academy. So not only can we apply it to very large-scale data, but simultaneously we can find incredibly diverse and subtle patterns inherent in this data that were not even intended. In this process, for both images and sequence like this, why did these two sequences appear consecutively? Why these three appear? Why these appear next to this? All this connect to protein functions. These patterns are found during self-learning even though they didn't intend it. This will be one very important aspect. Next, we encode this input data into vector form and transform it into more high-dimensional vectors. By high dimensionality, we mean dimensions increase to 100, 200, 1000 dimensions. So vectors are not just data. We transform data into vectors. Vector values are determined according to the extracted subtle patterns, and especially in methods with attention like those used in LLM or AlphaFold. This transformer line method captures dependencies between words or sentences and contextual information and vectorize them. As a result, when we map these vectors to some space in high dimensions, they are not mapped randomly but according to the context. For example, words related to animals cluster together and words related to fruits cluster together. So they are not mapped randomly. They are mapped in a state where relations between them are understood. We can do things like this. And what emerges from doing this is generalization and emergent intelligence. As I mentioned earlier, we create these embedding vectors, but because we create embedding vectors by understanding context, they can do various things, and one example of this is shown here. For example, we input English words like this, and we represent words in vector space, and although it is hard to see clearly here on the previous page, looking at it more clear here, if you take the vector for king, add the vector for man, and subtract the vector for woman, you get a new vector. When you convert that vector value back to a word, you get a word queen. So it's discovering relationships that can perform linear algebra between vectors. But this isn't just it, it's just one simple example, and it can do incredibly many things. When we classify documents, this is a paper published in Science by Professor Geoffrey Hinton, who won this year's Nobel Prize in Physics. Here, just by looking at documents, it can classify where this document came from without supervised learning. Here, just by looking at documents, it can classify where these documents come from without supervised learning. It classifies by itself without labels. The important thing is creating a meaningful vector space, and then as the scale of data and model scale increases, the vector dimensions increase, and by finding more sophisticated patterns, this vector space becomes more organized. This is extremely important, and from this organized structure, high generalization performance becomes possible. If I put small scale data into this, it maps onto the vector space I've already created and understands how this small scale data maps, and from that understands the problem. This could be considered through the origin of generalization performance. Secondly, as I mentioned earlier about similar things clustering together, these things cluster automatically without our intention during self-learning. As a result, it can understand context that people didn't intend. From that, show emergent intelligence and perform. Consequently, it can also develop various contents like creating videos from verbal descriptions, creating images, creating PowerPoint presentation, etc. I can do this, create things. Now, this process of foundation models mapping lots of data into high-dimensional data might seem, while not completely identical, similar to how people learn. For example, when you or I acquire new knowledge, we don't just store it on our head as is. Instead, we understand it through context and gain wisdom in a form that we can generalize. When we gain lots of knowledge, once we grab the principles of arithmetic, we can calculate any numbers given to us. This is also a human capability, and similarly, LLMs can do this with just a few examples of arithmetic understanding those relationships. It notices these things. And for example, with people, we know how to conduct ourselves in society. Looking at people, those with more experience know, I should treat this person this way and treat that person that way. Similarly, rather than remembering each person individually, we abstract in a way that generalizes. Naturally, this leads to certain biases. When we have lots of experience with certain content, and we receive new knowledge, we remember it in reference to our previous knowledge, which can sometimes lead to incorrect biases. LLMs actually have this tendency too. It varies depending on what data they've been more exposed to, and since we don't remember things like taking photographs, we just remember the essence and forget the rest. So our memories become distorted, and LLMs also sometimes provide so-called hallucinations, information that seem plausible but it is not correct. Looking at these things, I think they might be similar to how our brains abstract information. Next, I will talk about the present and future of foundation models. Currently, foundation models are much more developed in natural language processing and image fields. ChatGPT is one of the typical examples. It draws pictures well, handles various languages well, translates well. In life science, models called foundation models like AlphaFold or EEST4 or mRNA models exist, but they don't handle diverse types of data. Instead, they can deal more limit with integrated data. So while they can do structure sequence prediction well, they can't solve the various problems needed in bio and drug development. The generalization performance is still lacking because they haven't integrated diverse data. So in a way, I think what we are doing now is much at a baby steps level compared to what we expect. Currently, based on reductionism, as I mentioned earlier, both people and AI work with limited knowledge and experience suitable to their fields. So, for example, we solve problems by collecting bioactive data well, or applying bioinformatics by solving omics data well, solve structured data well, or handle ADM-related things well. These are all individualized. These predictions are all individualized in our HyperLab too. But solving individual problems well doesn't mean we solve the whole problem well. And we still can't understand the whole with just individual problems. For example, while we know AlphaFold does very well, not ChatGPT. In reality, AlphaFold doesn't solve all problems well. Recently, I saw one paper where they show examples of mutations that AlphaFold predicted. Here's a structure where mutation isn't possible. Compared to experiments, the RMSD value is less than 1 angstrom, so we can say that it's very accurate. It almost matches the experiment. It's quite amazing result. But what the authors of the paper did was they just randomly deleted residues around this pocket. As you can see in the slide, then what was here is gone like this. When asked to predict, the RMS is still 2 angstroms, 0.7 angstroms, 1 angstrom, looking here. And there are almost no residues, but it still predicts well like this. So to put it this way, it means that it doesn't understand biological or physical interactions. It just placing shapes properly. So it predicts even without residues. And even if I randomly mutated around these residues, the performance is still good like this. And this doesn't make sense. So AlphaFold might predict structures I've already matched as answers well, but it cannot predict these modified structures well. So we can see it's not easy to understand everything with just individual data. Because AlphaFold doesn't know what these long covalent interactions are, in this respect, we need foundation models, and we need to solve problems in a holistic way, not with just data, but integrating various kinds of data. By self-learning with this collected data, recognizing subtle patterns between them, even at levels people can see, and mapping them to vector space, we can solve various problems. Something like the virtual cell I mentioned earlier could be one example. If this is created, we can organize through zero-shot, few-shot learning, then do drug development that considers everything from target identification to clinical trials simultaneously. We can expect emergent function. Going further, a recent perspective paper in Cell talks about whether we could create AI scientists. AI could collect and organize information from various medical devices, and through this trained AI, human and AI scientists could collaborate on brainstorming, creating new hypotheses, debating certain research results. Since humans cannot know everything through this trained AI, we could use AI in various ways, like getting experts consulting in fields we don't know well. Having roundtables, even going further to self-driving labs, people talk about this a lot these days, where foundation models could perform many roles that people need to do in a self-driving labs, making drug development or bio research more efficient. This is how we expect the future might be. With this, I will conclude the webinar. The presentation was recorded version, and we will have a live Q&A session via chat for the following 20 minutes. So please feel free to ask about the presentation, and I'm happy to answer them. And I'd like to thank you for your interest in this webinar through this year. Thanks to you, our YouTube channel has already surpassed 1,000 subscribers. For those who haven't subscribed yet, please subscribe, and we will see you again next year with new content. Thanks a lot.
Yeah, so Hyperlab definitely made, everything a lot more streamlined, a lot simpler. I think all you needed to do was type in the PDB code, it would get it for you, and then you can draw in your ligand. Hyperlab is a lot, more streamlined in comparison to other docking methods. And then you can automatically go through and do the docking, do prediction of the different physical characteristics. I would recommend it if they, ask for something simple and easy to use, right? Especially if they don't have, the computational, know-how or the willingness to learn how to code on their own. So my name is Ryan Kern. I'm a 5th year grad student at Georgia Tech. And so I work in the oilary lab, and a lot of what we do is medicinal, so drug design, synthesis, just working on developing new candidates for drugs. Yeah, so Hyperlab definitely made, everything a lot more streamlined, a lot simpler. I think all you needed to do was type in the PDB code, it would get it for you, and then you can draw in your ligand. Hyperlab is a lot, more streamlined in comparison to other docking methods. And then you can automatically go through and do the docking, do prediction of the different physical characteristics. I'm the one person in my lab who does majority of the docking. Georgia Tech, we have a couple of computational groups who are much more into the weeds of they want to be able to control everything. We're mostly just interested in getting the thing in and then going into the lab and synthesizing it. It's a much easier ask to go, hey, will you use the software? Just one click, just, easy, straightforward. Asking them to use something like Hyperlab would be a lot easier to do than asking them to install Vina, run this Linux script, do all this stuff. So I mostly worked with the binding tool. Having the ADMT prediction, having everything in, a spreadsheet format where it has your structure, it has your predicted score, it has the predicted properties, helps keep everything organized. And I think it's just a very clean way to use it. And then I also like, when you're doing docking, you can also input standards, so known inhibitors with known inhibition, generate the graph where you compare the dock score with. And so I think that's a really cool feature. With the people who do, synthetic medicinal chemistry, there is always this worry that, well, you can't really touch or trust molecular docking. So having that to go, well, here you have this correlation, right? And so this should generally correlate with what we expect to see in the lab. I would recommend it if they, ask for something simple and easy to use, right? Especially if they don't have, the computational, know-how or the willingness to learn how to code on their own. It's definitely what I would recommend.
It's a really cool tool. You get really nice visualizations. It's really useful, really fast, can be really helpful at least for us. I am José Antonio Costoya. I am the head of the lab of molecular oncology in Thimus at University of Santiago de Compostela, and our main goal is to elucidate some oncogenic mechanisms with the purpose of finding new targets both for diagnosis and for therapy also. I mean, my background is mainly biomedical. I am MD and I'm not a chemist. It's crucial to manage all this big amount of information and be able to get the real important information for your work. Dealing in the lab, in the group, and with the doctoral fellows and PhD students, especially the people that it's getting their careers. All the information you have from ADMET, from the small molecules that you basically browse in your database that you are linked with your tool, is really useful, really fast, and be really helpful for at least for us. I think it's a really cool tool. I mean, as my first contact was at the conference, I feel that was an interesting tool for us because, first of all, I think it's quite important for this kind of tools, especially for the researchers in general, not the people that is working specialized in AI or in specific tools that is user-friendly. This, I think, is crucial. And yeah, of course, as a starting point is both from, especially for docking some targets that we have, proteins that we have, some small molecules, and was really cool. I mean, to see, to visualize is really, you get really nice visualizations. We are a wet lab that we need to use these tools, but we are not specialized on those tools. We are not focused only on those tools. It's really powerful, and we experiment that is still learning and growing, I guess. You still need experimental information for reach the final point.
Yeah, So HyperLab definitely made, everything a lot more streamlined, a lot simpler. I think all you needed to do was type in the PDB code, it would get it for you, and then you can draw in your ligand. HyperLab is a lot, more streamlined in comparison to other docking methods. And then you can automatically go through and do the docking, do prediction of the different physical characteristics. I would recommend it if they, ask for something simple and easy to use, right? Especially if they don't have, the computational, know-how or the willingness to learn how to code on their own. So my name is Ryan Kern. I'm a 5th year grad student at Georgia Tech. And so I work in the Oilary lab, and a lot of what we do is medicinal, so drug design, synthesis, just working on developing new candidates for drugs. Yeah, so HyperLab definitely made, everything a lot more streamlined, a lot simpler. I think all you needed to do was type in the PDB code, it would get it for you, and then you can draw in your ligand. HyperLab is a lot, more streamlined in comparison to other docking methods. And then you can automatically go through and do the docking, do prediction of the different physical characteristics. I'm the one person in my lab who does majority of the docking. Georgia Tech, we have a couple of computational groups who are much more into the weeds of they want to be able to control everything. We're mostly just interested in getting the thing in and then going into the lab and synthesizing it. It's a much easier ask to go, hey, will you use the software? Just one click, just, easy, straightforward. Asking them to use something like HyperLab would be a lot easier to do than asking them to install Vina, run this Linux script, do all this stuff. So I mostly work with the binding tool. Having the ADMT prediction, having everything in, a spreadsheet format where it has your structure, it has your predicted score, it has the predicted properties, helps keep everything organized. And I think it's just a very clean way to use it. And then I also like, when you're doing docking, you can also input standards, so known inhibitors with known inhibition, generate the graph where you compare the doc score with. And so I think that's a really cool feature. With the people who do, synthetic medicinal chemistry, there is always this worry that, well, you can't really touch or trust molecular docking. So having that to go, well, here you have this correlation, right? And so this should generally correlate with what we expect to see in the lab. I would recommend it if they, ask for something simple and easy to use, right? Especially if they don't have, the computational, know-how or the willingness to learn how to code on their own. It's definitely what I would recommend.
It's a really cool tool. You get really nice visualizations. It's really useful, really fast. Can be really helpful at least for us. I am José Antonio Costoya. I am the head of the lab of molecular oncology in Thimus at the University of Santiago de Compostela, and our main goal is to elucidate some oncogenic mechanisms with the purpose of finding new targets both for diagnosis and for therapy also. I mean, my background is mainly biomedical. I am MD and I'm not a chemist. It's crucial manage all this big amount of information and be able to get the real important information for your work. Dealing in the lab, in the group, and with the fellows and PhD students, especially the people that it's getting their careers. All the information you have from ADMET, from the small molecules that you basically browse in your database that you are linked with your tool is really useful, really fast, and be really helpful for at least for us. I think it's a really cool tool. I mean, as my first contact was at the conference, I feel that was an interesting tool for us because, first of all, I think it's quite important for this kind of tools, especially for the researchers in general, not the people that is working specialized in AI or in specific tools that is user-friendly. This, I think, is crucial. And yeah, of course, as a starting point is both from, especially for docking some targets that we have, proteins that we have, some small molecules, and was really cool. I mean, to see, to visualize is really, you get really nice visualizations. We are a wet lab that we need to use these tools, but we are not specialized on those tools. We are not focused only on those tools. It's really powerful, and we experiment that is still learning and growing, I guess. You still need experimental information for reach the final point.